The human brain is a complex organization of structures and pathways, and early investigators sought to discover the relationship between its structure and higher cognitive functions. The ventricular system was long given an elevated function, the fourth ventricle being in turn considered the seat of the soul, and later as the seat of memory. In the 18th and 19th centuries, the phrenologists took the idea of localized function to an extreme, producing maps relating areas of the cortex to such ethereal attributes as perceptiveness, wit and humor, and ideality. Given their rudimentary knowledge of the structure of the brain and of behavior, it was natural enough for these early workers, the phrenologists, to try to find one-to-one relationships between particular structures of the brain and descriptions of behavior that we now see to be quite inappropriate. By now in the course, you should appreciate that it's meaningless to try to say that a particular aspect of behavior is localized in a particular structure in the brain. For example, your listening to me talking now depends upon many regions of your brain being active simultaneously, and your understanding of what I'm saying depends not upon the functions of a single localized region, but upon the complex interplay between these many regions. Now, accepting that this is the case, then it implies that there are many neuronal interconnections between the various structures of the brain. And the significance of this internal wiring becomes most apparent when, unfortunately, something goes wrong with it.
All right, sir. Can you hold your hands up in front of you now for me? That's right. Keep them up there.
This patient is suffering from a disorder known as Parkinson's disease. It's believed to be caused by a malfunction in the extrapyramidal motor system, in a single pathway going from a region called the substantia nigra to the striatum.
You can, like that. There we are. Fast as you can. Now with the other hand. Fast as you can. Leave that one out of it for a moment. Hop.
The precise reason why a degeneration of this pathway results in involuntary muscle tremor and difficulty in initiating motor activity is not fully known. So little is at present understood of the internal wiring of the brain. But the ultimate answer to this and more general problems comes from a much more detailed understanding of the wiring of the brain. And it's in this context that I've come to the University of Oxford to talk to Dr. David Smith, whose work, along with his colleagues, has produced a significant contribution to an understanding in this area.
As you said, Jeff, in Parkinsonism, the pathway that degenerates is from the substantia nigra to the striatum. Now, if we're to understand how this degeneration leads to the loss of activity in Parkinsonism, the symptoms, we need to know which neurons in the striatum receive input from this nigrostriatal pathway, and secondly, where else in the brain they might send their projections. This is one example of a general problem in studies of the brain. That is, how can you show that three brain regions, distant brain regions, A, B, and C, are connected anatomically and functionally? By functional connections, I mean we have to be able to show that neurons in A, which send a projection to B, are actually in synaptic contact with neurons in B that project to region C. In order to solve this problem, we have combined three classical neuroanatomical techniques into one procedure in one animal. The first technique we used is Golgi staining. Now, if you take a brain section from a rat without staining it, then it simply is impossible to study the detail of the neurons in the light microscope. What Golgi found was that if you actually impregnate pieces of brain with silver salts, then you can produce sections which look like this, and this is because many of the neurons have become impregnated with the silver salt. And if you look under the light microscope, what you actually see, in this case in the striatum of the rat, you can see an individual neuron here. Its cell body is impregnated with the silver salt, and coming out from the cell body, several dendrites. And the silver stain impregnates the whole of the dendritic tree, if you're lucky, including, as you can see up here, some of the dendritic spines. Now, also in this picture, you can see some very thin processes impregnated. These are axons. They don't probably belong to the same neuron. So you can actually impregnate axons with the Golgi method, but the problem is you can't trace these axons over long distances in the brain. So the Golgi method is very beautiful at showing up the morphological type of a neuron, its dendritic tree and characteristics, but can't tell us where it projects. For this, you have to turn to other methods, and one way of studying projections to distant brain regions is to place a lesion in one region, which kills some of the neurons, and that leads their fibers and nerve terminals in a second region to degenerate. You can study this in the light microscope and in the electron microscope, and we chose the electron microscope because it allows us to study the synapses. Now, this is a high-power electron micrograph, about 100,000 times magnification, of a tiny part of the striatum. And here is a normal nerve terminal with its characteristic synaptic vesicles. It's in synaptic contact with the dendritic spine here, and we know it's in synaptic contact because of this postsynaptic thickening, dense material. Close by, we can see some more postsynaptic thickening in a spine, and presynaptic to that is a degenerating nerve terminal, which we can identify because it is dark, that is electron dense. In this animal, a small lesion was placed in the cerebral cortex, and so this shows that neurons in the cerebral cortex send fibers which form terminals in the striatum on dendritic spines. Now, although this is a very powerful method, the very power of it depends upon the high magnification and the ability to see the synapses. It is therefore very limited because we cannot see from this sort of picture what type of neuron receives this synaptic input. We can't possibly say which neuron the dendritic spines came from. The third technique also traces connections, but relies on the dynamic living properties of the neuron. In this case, we are studying the retrograde transport of protein from the nerve terminals back to the cell body. If you study a neuron, you find that the nerve terminals are constantly sampling material from the extracellular space and transporting them retrogradely back to the cell body inside small vesicles that travel along the axon. If you introduce a marker substance, we used horseradish peroxidase, then this is taken up in the same way at the terminals and transported back to the cell body. So if you introduce horseradish peroxidase into one area, and then about a day later look in the second area, you can find cell bodies containing the enzyme after it's been transported retrogradely from the nerve terminals. In this way, we can establish a connection between these two areas. When you look at cell bodies, which have been labeled in this way by horseradish peroxidase, in the microscope, in the light microscope, you can see the blue reaction product filling the cell body. And this is a typical section showing such cell bodies with horseradish peroxidase in them. One thing you will notice about this section, however, is that you can't see the dendrites of these neurons. And these neurons are in the striatum, and we know they probably have dendrites, but the horseradish peroxidase does not penetrate into the dendrites. So this is a very powerful method for showing connections between two brain areas, but unfortunately it tells us nothing about the type of neuron which is projecting.
So David has now introduced three classical techniques. Firstly, the Golgi stain, which stains almost complete cell bodies, and so we can identify what type of cell they are. Next, lesions in a particular structure produces degeneration, so that we can identify degenerating synaptic terminals. And finally, horseradish peroxidase injected into a structure is taken up by the synaptic terminals and transported back to their cell body. And now remember what David Smith's problem was. He wanted to confirm that neurons in a particular structure, and let's call it structure A, projected to another structure, structure B, and that the neurons from A made synaptic contact with neurons in structure B, which in turn projected to a third structure, structure C. And here's David Smith again.
Well, what we do is place a lesion in region A, then by looking for degeneration in terminal B, we can identify dendrites in region B which are in synaptic contact with the degenerating terminals. But of course in the electron microscope we cannot identify the neurons to which these dendrites belong. We then inject horseradish peroxidase into region C, which retrogradely labels cell bodies in region B. But there is no proof that the dendrites in B which receive the input from A actually belong to the cells in B that project to region C. In other words, we still haven't demonstrated a connection between region A and C. To solve this problem, we thought it would be nice if we could stain these HRP-labeled neurons by the Golgi method. And then, if we were lucky, we might see one of their dendrites in synaptic contact with a degenerating bouton due to a lesion in A. However, until now, it's not been possible to combine Golgi staining and horseradish peroxidase tracing in the same animal. Technically, this proved to be a very great problem, but it was solved in 1978 when Peter Somogyi from Hungary came to work with me. Together with a young biochemist, Anthony Hodgson, they found a way of demonstrating horseradish peroxidase in brain slices which were thick enough for Golgi staining. Thus, at last, it seemed as if we'd got somewhere. We might actually be on the track of something important.
And in fact they were. Now you can see the contribution that the group made. In putting together these three techniques in the same animal, they were able to positively identify synaptic terminations and also the nature of the cell on which the terminations were made. Now, the specific problem the group worked on was the details of the interconnections between structures in the extrapyramidal motor system. And these are the cortex, the caudate nucleus, the putamen, the substantia nigra. Now, next I spoke to Dr. Paul Bolam, who'd used these three techniques to look at a specific pathway running from the cortex through the striatum to the substantia nigra.
The pathway that I was interested in was that from the cortex, that's area A, through the striatum, that's area B, to the substantia nigra, that's area C. Now, in the rat, the main projection area of the striatum is the substantia nigra. And we wanted to see whether these cells going from the striatum to the nigra, that's the striatonigral neurons, received a direct synaptic input from cells in the cortex. The way we did this was to first place a small lesion in the sensory motor cortex of the rat while it was deeply anesthetized with chloral hydrate. 24 hours later, we injected a very small volume, about 50 nanoliters, of a solution of horseradish peroxidase. After a further 24 hours, the striatum was removed and cut into slices of about 1 millimeter thick. These slices were then incubated to reveal the retrogradely transported horseradish peroxidase, and they were then Golgi stained. This part takes about 4 to 5 days, and by this stage the slices have turned black. So that we can now see the stained cells in the light microscope, we have to resection these blocks at about 100 micrometers. And we then put them onto microscope slides like this and search through them in the light microscope for cells that are both Golgi stained and retrogradely labeled with horseradish peroxidase. Now I have on this slide one of the cells that we found in the striatum. If we look at it under the light microscope, you can see that the lower right of this cell body is stained an orange color, and this is the HRP reaction end product. So we can immediately tell that this cell projects to the substantia nigra, that's area C. But if you remember, the HRP only stains the cell body and not the dendrites. But if you look at the top left of this cell, you can see it's stained black, and this is the Golgi deposit. If we now change the focal depth, you can see the dendrites come into focus. And so we can now immediately morphologically classify the type of cell in the striatum, that's area B, that projects to the substantia nigra, area C. As I mentioned earlier, this same animal received a small lesion in the cortex, and this should result in degeneration occurring in the striatum. But the only snag is we can't see the degeneration at the light microscope. So we have to take this same cell and examine it in the electron microscope and look at its dendrites to see if there are any degenerating synaptic terminals. Now, for the electron microscope, we need sections which are about 1,000th as thick for the light microscope. And to do this, we have to cut the cell out of the section on the microscope slide and re-embed it in a block of resin like this. And in this state, it's ready for sectioning on the ultramicrotome. Now, this machine is specifically designed to cut sections thin enough for the electron microscope, and we probably get about 500 for each cell. Now, each of these thin sections is then looked at in the electron microscope. We locate the dendrites which we've seen in the light microscope and search along them for any terminals which are degenerating. Now, I have some results from this same cell here. And the first thing we have to do is to prove that we're looking at the same area as we've already seen in the light microscope. And this photograph is of the Golgi stain section. And here you can see some of the Golgi stain spiny dendrites. And these belong to the HRP labeled neuron. And this photograph is a low-power electron micrograph at approximately the same focal depth and about the same magnification. Now, this, unfortunately, is a fold in the section. It always seems to happen on the best sections. Now, we know we're in the same area because we can use landmarks like neurons, unlabeled neurons. And in the light micrograph, we have 3 unlabeled neurons here, here. And they also appear in the electron micrograph here, here, and here. And similarly, we have 3 capillaries in the light micrograph here, here, and here. And they also appear in the electron micrograph here, here, and here. Now, this dendrite was particularly interesting, and it also appears in the electron micrograph here, but it's only just visible at this magnification. And so we looked at this area at a higher magnification. And this is in this photograph. So we can see this same capillary here. And here's the main dendritic shaft. And we can recognize it because of the black Golgi deposit. And at this point, a spine emerges. And if we look at this area at a higher magnification, we can see the main dendritic shaft, the dendritic spine, and here is an axon terminal making synaptic contact with the spine, and here's the postsynaptic density. Now, this terminal is showing the characteristic signs of degeneration that David talked about earlier. And because in this animal we placed a lesion in the cortex, we know that cells from the cortex, that's area A, are in direct synaptic contact with cells in the striatum, area B, and that these cells project to the substantia nigra, that's area C. So our results prove conclusively that there's a monosynaptic link between the cortex and the substantia nigra through the morphologically identified cells in the striatum. Now, this, of course, may not be the only pathway between these 2 structures, but at least demonstrates one of them.
These are very important results because they show that the cortex can influence the substantia nigra directly through a relay in the striatum. The next problem we studied in relation to the control of motor activity was whether the striatum can influence the thalamus. As you know, the thalamus is an important relay to the motor cortex. Classical methods had shown that the substantia nigra projects to the thalamus. And what we wanted to find out was whether the striatum can influence the thalamus via a relay in the substantia nigra. In this case, we applied the same techniques as before, but here A is the striatum, region A is the striatum, region B is the substantia nigra, and region C is the thalamus. We placed a lesion in the striatum, we injected horseradish peroxidase in the thalamus, and we stained the substantia nigra by the Golgi method. We were able to show that, in fact, neurons in the substantia nigra that project to the thalamus do receive input from the striatum. In other words, we have yet another link in the chain. If we put all of these results together, we get part of a circuit by which the striatum might influence motor activity. First, as Paul showed, the cerebral cortex sends a projection to the striatum, to neurons which themselves project to the substantia nigra. Then, as I've just mentioned, neurons in the striatum project to neurons in the substantia nigra, which themselves project to the thalamus. And it's well known that the thalamus sends a projection to the motor cortex. What we're doing at the moment is studying other influences on the striatum. It is known that the substantia nigra projects to the striatum, and this pathway could modulate any influences on the striatum that come from other regions. For example, we are looking at the possibility that the limbic system, which is connected with our internal states, our feelings and our emotions, may have an input into the substantia nigra, and so could, by this route, influence our motor activity. We might then get a picture of the striatum as a kind of integrator, which receives input from sensory systems via the cerebral cortex, that is, our external world, but also from parts of the brain concerned with internal states, the limbic system and the substantia nigra. So in this way, we can begin to see how an animal might balance his internal needs and motivations with information he receives from the external world and convert this into an effective motor response. In fact, going back to the beginning with the Parkinsonism patient, you remember that here the nigrostriatal pathway is ineffective. And so we could speculate that this may be the reason why this patient can't convert his desires into motor activity. So by unraveling pathways in the brain, we're beginning to get a very important insight into our whole control of behavior. Furthermore, it is giving us a rational basis for the design of drugs to treat diseases like Parkinsonism and others related to it, including perhaps mental illness.